We're good now. Welcome everybody. It's good to see you all here for our last installation of our fall 2020 Living with Wildlife seminar series. This is when we basically are opening up our Living with Wildlife class to the university community to let people come in and take advantage of the wonderful set of speakers that we've gathered here to share information with us. So today, before we get started with our speaker, I just want to make one quick acknowledgement. So this is something that we like to do whenever we gather, even though we're gathering virtually and we're all in different places. I think it's still a good time to do this. So we recognize the University of Wisconsin-Stevens Point occupies lands of the Ho-Chunk and Menominee people. Please take a moment to acknowledge and honor the ancestral Ho-Chunk and Menominee land and the sacred land of all Indigenous peoples. All right. So with that, I'm going to stop sharing my screen and I'll introduce our speaker today. So Dr. Marie Perkins is here with us. She is our newest faculty member in the wildlife discipline here at UW-StevensPoint. And her specialty is actually in toxicology and in mercury, as you might have guessed from the title of the talk today. So I'll let her say a little bit more about her background, but I just wanted to say on a personal note that Marie is one of our most favorite members in the wildlife discipline because of the zest that she really brings for her teaching. But it's also really endlessly fascinating to me. I don't know if I've ever told you this, Marie, but it's endlessly fascinating to me to really get to talk to you about the work that you do. And I've really been looking forward to this this presentation, perhaps most of all. So thank you very much for sharing your time with us tonight. What we're going to do is let Marie go ahead and share her screen with us. And then if you have any questions as we go through the talk, if you just chat them in, just like we've always been doing, we'll get to those questions at the end of her presentation.
All right, thank you very much for the introduction. I'm really happy to be here today to talk to you guys about mercury. I don't really have much planned to say about myself, but I just started here as a professor in wildlife ecology just about a year and a half ago now. So I'm a relatively new professor here, but have been enjoying it so far. The research that I did for both my my PhD and my postdoc really focused in on mercury and wildlife, and specifically in birds, with a little bit of human health mixed in. So one of the reasons that I really am excited to talk to you today about mercury is that it's actually really fun to talk to people about mercury, because kind of everybody knows a little bit about mercury, right? So maybe you've seen signs at, a lake that you, or a river that you fish in that, is giving you some guidelines for eating the fish that you catch. Or maybe you've seen this seafood watch list kind of indicating better fish choices to reduce your exposure to mercury. Or as you guys were coming in, I was talking to Scott about, having that liquid mercury and people playing with it back in the day, which no one recommends you do now. The other reason why I really like to talk about mercury is that even though it's a pretty well-known contaminant, and it's known that it negatively has some pretty bad health effects, there's still really a lot about mercury and mercury exposure in both humans and wildlife that we don't know. So it ends up being just a really interesting area of research, I think. So what is mercury? It's this global pollutant that can pose a really significant health threat to humans, wildlife, and ecosystems. And so if you guys have caught any of our wildlife health seminars that have been going on over the past month or so, I think almost every speaker has brought up this idea of One Health, which is that this combination of human health, environmental health, and wildlife health, and kind of the importance of all three for just health overall. And those speakers have focused kind of mostly on zoonotic diseases, related to pathogens or epidemics and pandemics. But One Health really fits well in the idea of health related to pollutants and contaminants and toxicology. And so I really think that mercury is a really good example of a contaminant that, you know, the human health, environmental health, and wildlife health are all tied together. So kind of throughout the talk, I'm going to kind of tie these different pieces together for you. And so just a bit more background on mercury. So it is this metallic element that's liquid at room temperature, which is why it has a lot of really interesting qualities that have made it really useful for humans. We have these two forms, which are the inorganic mercury form, which is kind of that liquid metal. And then we have organic mercury, which I'll talk a bit more about going forward. But we'll start with our sources of inorganic mercury. And so we have some of these natural sources. So particularly mercury can get released into the atmosphere from volcanic activity. But these natural sources tend to be pretty minor in the scheme of things. And so the majority of the sources come from human activities. And so we know that mercury has been used in a variety of consumer products. So we've got our mercury thermometers there, which no one should have anymore. We should get rid of all of those. But we do know that there's an amount of mercury in fluorescent light bulbs and various other consumer products. It's also used for a lot of different industrial processes. And there was a lot of historic point source contamination where industries back, you know, in the early maybe 1900s, late 1800s, dumped mercury out inorganic mercury out into the environment. And those contaminations persist through today. But really the majority of mercury from human activity is atmospheric emissions of mercury that come from coal burning power plants and from small scale gold mining. And so we know that, you know, since about the mid 1800s, mercury emissions from human activities has really dramatically increased. And so this graph is just, you know, we've got the time scale on the x-axis and releases of mercury on the y-axis. And we see that even now there's quite a bit of mercury releases coming from various areas, particularly Southeast Asia. And so we can really see how mercury is referred to as a global contaminant. We see that these are, you know, mercury emissions across, which are really widespread across the globe. And so these are atmospheric emissions. And once mercury is released in the atmosphere, it can really travel these long distances and be deposited on the landscape quite far away from the source. Once mercury is released in the atmosphere, it can remain up in the atmosphere for about 6 months and just travel related to, you know, wind currents. And so that's, you know, when we're thinking about this form of inorganic mercury, but once that inorganic mercury gets deposited across the landscape, it can then be converted into our organic form of mercury, which the main form we talk about is methyl mercury. And methyl mercury is the more toxic form of mercury. And it's the form of mercury that is much more available to humans and wildlife. So it's the form that's really prevalent within food webs. And inorganic mercury gets transformed into methyl mercury by bacterial processes. And this occurs most readily under anaerobic conditions. And so we see these higher levels of methylmercury in areas that are, wet or aquatic, so wetlands and lakes and river systems. And so we often see that mercury exposure is more prevalent in these landscapes. And because methylmercury is the form of mercury that's more available within the biota, when we talk about mercury exposure in humans and in wildlife, we're almost always referring to exposure from methylmercury. So in addition to methylmercury being more toxic and more available within biota, it also increases up the food web. And so we end up having these top predators that can have really high concentrations of mercury. And this is one of those reasons why we have these kind of different fish consumption guidelines, because fish that feed higher up on the food web tend to have these higher concentrations of mercury. All right, so much of my research really focuses on mercury exposure, and both humans and wildlife can be exposed to mercury through their diet. And I really think that, looking at mercury exposure, my dog is coming in the room. I forgot to close the door, sorry. Anyway, looking at mercury exposure is, important for quite a variety of reasons. One is that exposure to mercury is really, detrimental to the health of both humans and wildlife. So in humans, humans are most often exposed to mercury through fish, so through eating fish, which is why we see all these consumption warnings. And it can have a really detrimental effect on human health. It's a neurotoxicant, and so it can negatively affect the brain and the nervous system. And this is particularly true for developing brains, so those of fetuses and children. And so we see that, exposure, even low exposure to mercury to fetuses can result in decreased IQ scores, and that this really relates to real costs in society. So there's, they've put some, price estimates on the dollar amount lost to reductions in IQ due to exposure to mercury. Mercury is also really bad for wildlife. So when I first started working and researching mercury, I kind of knew that it was not good for humans, but it just hadn't really occurred to me that, it would have these negative effects on wildlife as well. And so, there's been some research looking at the negative health effects on mammals, and they can negatively affect their reproduction, physiology, behavior, neurochemistry. And so there haven't been a lot of studies looking at mammals, but all of these studies indicate that it can really negatively affect mammals. There's been a lot more research looking at the health effects of mercury on birds, and this is also the area that I have focused on. And we see that it has just a whole variety of negative health effects. So it can really affect reproductive success, reducing the number of eggs that hatch or the number of nestlings that fledge. It can change the song. So little, male birds out there just kind of like singing his heart out, but the females are like, yeah, I'm just really not interested in that, So that can also have effects on reproduction. It can affect flight, have a negative effect on the immune system. And there's been, studies that indicate that these negative health effects can have these kind of population level that might be resulting in population level effects. So these kind of combined health effects may be one of the reasons why certain bird populations are decreasing. or declining. So mercury is bad for health, right? So it has these negative health effects. So we want to look at mercury exposure to understand, how this might be affecting health, but particularly looking at mercury exposure in wildlife, we often use wildlife as bioindicator species of mercury within the environment. And this is because the global mercury cycle is really complicated. So just looking at these emissions or depositions doesn't really give us the full picture, right? Because we've also got we've also got this added methylation going on that really would influence exposure. And so a lot of times we look at wildlife species as indicators of where mercury exposure risks are going to be the greatest. And so we can kind of think about this as like the canary in the coal mine idea of using an indicator species to tell us where we might be having areas that might have negative effects on human health as well. And so birds have been used as bioindicators of mercury exposure, and they tend to be pretty good bioindicators because they're relatively abundant, they're widespread, they incorporate mercury into their tissues pretty readily, and they're easily sampled. And in particular, we can collect blood and feathers without actually harming the individual birds or having any sort of effect on the population. Look at the concentrations in those tissues and get an indication of their exposure. And so this is just an example of a study that really looked at mercury concentrations of blood in a whole bunch of different bird species all across the western half of the continent. And you can see the blue color are going to be lower mercury concentrations, where red colorations are going to be higher mercury concentrations. And so looking at these concentrations in birds can give some really good indication of areas that might have high mercury contamination and might be a risk to other wildlife as well as to humans. So just, that's kind of a big background about what mercury is, how it's kind of moving within the environment, and kind of tying in human health and wildlife health. And so I really want to kind of focus in a little bit on the Arctic as kind of a case study and to give you some more specific examples. So we often think about the Arctic as being kind of this pristine environment, right? But really, because of wind currents and ocean currents, as well as really these kind of unique conditions that occur in the Arctic, mercury tends to get deposited in the Arctic at much higher rates than it gets deposited in other locations. So again, the mercury is not really being produced in the Arctic, but because mercury is this global contaminant, it gets transported and deposited across the Arctic landscape. And we do see that wildlife within the Arctic tend to have high mercury exposures. There's lots of studies on lots of species that show these high mercury exposures. And so these are just some examples of adorable megafauna, as well as, a fish that have been shown to have high mercury concentrations in the Arctic. And not only do we see that, we have this concern for these species, right, and the negative health effects that could be coming, just for these Arctic wildlife, but many of these species are also country foods that our native Arctic communities are eating. And so we do also see that the community members of these Arctic communities also tend to be exposed to mercury in higher concentrations than some other areas. So this is, sorry about the figures, a little bit small, but generally it's blood mercury concentrations of women in these Arctic communities. The larger circles indicate higher blood mercury concentrations, and then the darker coloration are more recent samples. And we do see that there is cause for concern of mercury exposure to within these populations. And so to tie in some of my own research, and also why I thought I'd talk a bit about the Arctic, is for my PhD, one of the projects that I worked on was looking at mercury exposure in Arctic breeding shorebirds. And we wanted to look at mercury in these shorebirds for a couple of reasons. One is that they are good indicators of terrestrial mercury. So during the breeding season, they're up there nesting and foraging in the terrestrial landscape. So a lot of mercury concentration or mercury exposure we know in wildlife are marine associated or fish eating species. And these shorebirds are terrestrial. And they all kind of have this similar diet of insects. So we often think, again, about mercury exposure being related to eating seafood or fish, right? That's how humans are often exposed, and a lot of wildlife are exposed from eating fish as well. But we do know that mercury moves within these terrestrial systems and within these invertebrate systems as well. And some invertebrates can have a pretty high mercury concentrations. So we wanted to look at these insect-eating birds. One of the other reasons why we wanted to look at shorebirds is that these Arctic breeding shorebirds are suffering pretty dramatic population declines. And so we were wondering if maybe mercury might be one of the factors that might be related to these declines. So overall, I wanted to establish current mercury concentrations in these shorebirds. So we had 9 study sites across the North American Arctic, 5 in the US, in Alaska, and 4 in Canada. We wanted to look at what some of the factors that might influence these mercury concentrations are, and to identify what some of the species and regions of risk might be. So using these birds as bioindicators of mercury in the terrestrial Arctic. We collected feathers and blood from these birds, and then they were analyzed for total mercury concentrations. We had 11 species of shorebirds that were sampled during the study, and the majority of these are of high or moderate conservation concern. So we sampled a lot of birds and a lot of samples. So overall, we had almost 2,500 blood and feather samples from our sites. And I'm going to focus mostly on the blood mercury concentrations. And this is because the blood mercury concentrations are going to be the most representative of exposures in the Arctic. And we analyze these for total mercury concentrations, but as I kind of mentioned earlier, these tissues are majority methyl mercury. So we're really looking at that methyl mercury exposure. So some of the results that we found, and again, we've got, oh, sorry, our study site of Barrow, the city of Barrow, went through a name change like over the course of the research project. And so I was trying to like get the new name up there, which I cannot pronounce. So I'll still refer to it as Barrow, but I really want to highlight that it should not be called that any longer. It's gone back to it's a more original name. So we overall found that the mercury concentrations in our shorebirds across the Arctic were relatively low. So that was surprising. We thought we were going to find high mercury concentrations kind of across the Arctic, but we did see that a couple things really stood out. Oh no, wrong way. There we go. One was that we saw really pretty high mercury concentrations across all of the shorebird species that were sampled at the Barrow study site. And then conversely, at its very nearest study site, the Utqiaġvik study site was where we saw the lowest mercury concentrations. So they were very, very low in the species that were sampled there. So, you know, this just is giving us some indication that there definitely are some areas within the Arctic that might be more at risk for mercury contamination and mercury exposure in wildlife. And so for an example of that is that for one of our species, the Semipalmated Sandpiper, they had about 6 times greater blood mercury concentration at the Barrow study site than at the Utqiaġvik study site, which I think is only about 100 miles away. They're pretty closely located to each other. One of the other really interesting findings that we had were that we saw these really pretty dramatic differences in blood mercury concentrations among the different shorebird species at the same site. So again, as I mentioned before, they're all kind of foraging on really similar foods. They're all foraging on these invertebrates or insects. They're all kind of located near each other. So they all kind of nest in the same area and are kind of foraging in the same region. But they have these really pretty different mercury concentrations. And we really did see this. So I'm just giving you the Barrow study site here because it's the kind of most obvious graphic, and we had the most species sampled there. But you know, we really found these pretty dramatic differences, right? So we've got about 4 times greater mercury blood mercury concentrations in our long-wheeled Dowitchers compared to American golden plover. But one of the things that we also found was that some of the species were pretty consistently high across study sites. So at all of the study sites that the Semipalmated Sandpiper was sampled, it had high mercury concentrations compared to the other species. And the same thing with our Dunlin and our American golden plover. So at all the sites that they were sampled at, they also had some of the lowest blood mercury concentrations. And so this was a really interesting finding and also gave us some good information about using these bird species as bioindicators that really which species you select to use as a bioindicator species could really give you a different idea of what's going on with the mercury at that location. So for example, if we had only sampled American golden plover, we would have come to the conclusion that mercury concentrations and exposures were low all across the Arctic. So having these multiple species really gave us a lot of information. We also looked at some differences in sex and generally found that males had higher mercury concentrations than females. And some other bird species, this has been shown to be true as well. And this is because females can deposit mercury into their eggs. So as they're laying eggs, they can excrete mercury into those eggs. And so this can sometimes result in lower blood mercury concentrations in females. And so in general, we found these low concentrations, but what do they really mean for the health of our Arctic breeding shorebirds? So we don't have specific concentrations that are indicative of risk specifically for Arctic breeding shorebirds. And we know that these kind of risk benchmarks can really differ among bird species. But just kind of using some of the information that we know about other kind of similar species, so in smaller insectivorous birds, we put together these kind of benchmark categories of risk. So we can kind of see that blue coloration and purple coloration are going to be kind of no risk or low risk. And then we get kind of into that orange and red are going to be kind of a moderate to moderate high risk of these negative health effects. And so in general, our Arctic breeding shorebirds were mostly at pretty low risk of negative health effects, but we did see that certain locations were likely to have greater risks, and that certain species were at higher risk as well. So overall, our study showed that these mercury concentrations, we were able to establish mercury concentrations across the terrestrial Arctic. We had about 1,100 blood samples that we were able to incorporate into kind of getting these current concentrations, which can then hopefully be used in future studies to look at whether or not exposures are increasing over time. We did see that our blood mercury concentrations differed by study site as well as by species, and also by sex. I didn't add that one in the summary, but we saw that as well. And that overall, our Arctic breeding shorebirds were at low risk of having these adverse effects, but that certain species were definitely likely to be at much higher risk. So I feel like we've got some not too bad news for Arctic shorebirds, which is good. But in general, it's kind of this doom and gloom presentation, right? Of there's lots of mercury being emitted into the atmosphere, and we see a lot of mercury exposure in our wildlife, in humans, and in these areas that we maybe would think of as being pristine. But there's some good news on the horizon as well. So there's been a lot of worldwide efforts to reduce mercury emissions from human sources. And specifically, I want to talk a bit about the Minamata Convention on Mercury. So this is a global treaty that aims to reduce mercury emissions across the globe. So any of the countries that ratify this treaty, and right now I went today and got the most updated numbers on this. So it looks like we've got about 125 countries that have ratified the treaty, including the US. We were one of the early signers, ratifiers of the treaty. And so once a country ratifies this treaty, then they are kind of beholden to a set of regulations which are going to aim to reduce new mercury emissions. And one of the parts of the treaty looks at determining current mercury exposures in both humans and wildlife, and looks at ways that we can determine whether or not the regulations that are implemented under this treaty are actually reducing mercury exposures across the globe. And so a lot of my research really focuses on aiding the implementation of this treaty and kind of establishing current mercury exposures that can then be used in the future to look at any of these kind of whether or not these regulations are actually helping to reduce mercury within the global system. And so this is a pretty exciting treaty. It's been ongoing, I think, since 2013. And so countries now are really starting to implement these regulations. I think the one when I kind of went to the website, one of them was looking at consumer products. So one of the portions of the treaty is to no longer be producing consumer products that contain mercury. So that one's pretty excited. It should be, I think, implemented in 2021 for many of the countries. So there's some really good hope on the horizon for reduction in mercury emissions globally. One of the downsides is that mercury is really persistent within the environment. Once it kind of gets into the system, it can last a long time and continue to cause exposures within the food web. This, my last slide. Yep. So with that, I'm a little short on time, but it looks like we might have a lot of questions. So that would be fun, I think, to have some time to answer some questions.
All right, we did have a number of questions come in. Thank you, Marie. That was awesome. So I guess I can ask you, would you prefer to go through the questions yourself one by one? Do you want me to read them to you, or do you want me to unmute people to let them ask their own questions?
Oh, I would say I would like them to be read, or people could ask their own questions. I don't know what people would prefer. I don't want to read them.
Okay, that sounds perfectly reasonable. Okay, so I tell you what, we'll go ahead and we can unmute people. What we'll do though is we will start at the top, and so I think that well, I tell you what, it looks like there is actually a question from a student that I might prioritize over like my repeated questions and Scott's questions. So maybe we'll let Dan ask your question first, and then if you guys, if anybody else wants to put your questions in the queue, go ahead and type them in, and then we'll let you ask your question once we get to it. Does that sound good this time? I'm looking at Dan. So Dan looks like it's okay. All right, go ahead and ask your question, Dan.
All right, this is verbatim. Is there a reliable method for accumulation and sequestration of ambient mercury that's being implemented now that you are aware of?
Not that I'm aware of. There's certainly like some ideas out there. There's nothing that I know of that's like any sort of like large scale idea of trying to remove mercury once it's already in the environment. One of the best things for mercury really has been the Clean Air Act. So because a lot of mercury is coming from coal burning power plants, all of the regulations and kind of scrubbers and technology that's been added to these coal burning power plants to meet the criteria for the Clean Air Act also reduce mercury. So that is part of the reason why we see a lot of emissions in Southeast Asia, particularly India now. They just don't have a lot of these technologies on their coal burning power plants. Although China now has been implementing a lot of those, which should help and reduce some of these emissions.
Okay. Are we kind of just relying on them, like the microbes and decomposing material to like bring it into the soil and just not get disturbed?
Yes, that is true. So basically the way that we get rid of mercury is to bury it. Yep. So for it to get, for it to get buried, especially in these waterways, buried in the ocean, although there's some recent evidence that that's just coming right back up. Yeah, and like you said, not disturbing them. So a lot of times if you have to go in and dredge or disturb lake sediments or river sediments, you're really just redistributing that mercury back into the system. Yeah.
Thank you.
Yeah. That's also an issue in the Arctic. I'll point out that melting permafrost is really expected to release a lot of mercury into the Arctic that has been, you know, buried over the last century or so.
That's a happy note.
Yeah.
Awesome. So my first question that I had for you relates to kind of what you were talking about with Dan. I asked, what's your take on clean coal? Like, I think a while ago we heard a lot about clean coal. Just, do you have a personal and or professional opinion about that?
Well, I guess I'll give both. Obviously, adding anything we can do to reduce the pollutants, mercury and other pollutants that are coming out of burning coal is better than not doing it, right? So the more that we can, you know, reduce the air pollution that's being produced from coal is an improvement. But I think, personally, I think reducing our need for burning any sort of fossil fuels, coal or otherwise, is really one of the only ways that we're gonna really reduce our mercury emissions. So I think, yeah.
So maybe not clean coal, but cleaner.
Yeah. Yeah. I think, I mean, you know, one of the things that we really did see in North American and also in Europe is once these, you know, Clean Air Act policies were put into place, mercury emissions really dropped. And now we see a lot more in these areas that don't have those policies instituted. And again, because mercury is a global pollutant, that mercury still makes its way back over to North America. But so yeah, the cleaner we can do it, the better.
Okay, fair enough. Scott, I think we'll go to your question about Greenland.
Yes, sure. I was curious, in one of the images you showed, Greenland had lots of spots on it indicating high levels of mercury. I was just curious the sources of that mercury, if it's just native or industrial, commercial, or blown in or what?
Yeah, I think a lot of it is coming from this global system. So it's just being deposited from this these releases of atmospheric mercury. A lot of it also comes from what they're eating, right? So a lot of times when we're thinking about eating marine mammals, those marine mammals are traveling, so they're getting the mercury from the marine system in a variety of locations, right? And then they're being consumed in those countries.
I was reflecting on one of our professors in biology, Bob Rosenfield, who does field trips with students up to Greenland quite often and does work with gyrfalcons and peregrine falcons, and they're being at the apex of the predator pyramid. You can imagine what mercury levels are there and what kind of impact it might have on those populations, behavior, and all those other effects.
Yeah, I would expect that those probably do have pretty high mercury exposures.
Yeah.
Scott, I think you've got one more question in here, and then after this, I have a question for our students.
Yeah, I was curious, and this one reflects back to the students too. I'm kind of curious if they're familiar with the history of the Wisconsin River, because it's right next door to us. At least if you're on campus, it's right next door. And the history we've had with paper mills, with pollution in the river, with high mercury levels, impacts on fish, water quality. There's quite a history there. And the Wisconsin River has often in the past been referred to as the hardest working river in the world. And it's because of all the damming and the paper mills and sawmills. I was just curious if today's students really have an awareness of that history that we have going on right next to us. So give me a thumbs up if you're aware of the issues that have happened in mercury levels in the Wisconsin River. You've got your little thing there, the reactions thing, this one.
We've got a few.
All right, we have a few, but there's a long history. Maria, I know you've not been here long. We've talked about it a little bit. Have you familiarized yourself with the Wisconsin River history?
Not a little bit with the history. I definitely know that it is not a clean river from a contaminant standpoint. And again, like I mentioned previously, these old pollutant sources, so for mercury, particularly from these paper mills and things, that persists. Those can persist, up at least 100 years and still be incorporated into the fish or into the be transferred into the terrestrial food web. And yeah, so I definitely am aware that the Wisconsin River has a lot of pollutants, not only mercury, but also organic persistent pollutants as well.
Mm-hmm. For decades, mercury has been used as a fungicide. And when pulp mills have logs brought in, they would oftentimes spray those logs with mercury to retard the fungal growth, because the fungus would stain the pulp, which would then stain the paper, and they wanted nice bright white paper. And so, all that processing, all the byproducts from those paper mills on the river back 30, 40, 50 years ago, a lot of it was dumped back in the river. And so a lot of it's industrial sources. And then that got into the food chain and into the fish. And then we had all kinds of fish consumption warnings from the Department of Natural Resources as far as the river goes. And fortunately, the Clean Water Act, back in the late 1960s has had a huge impact on the Wisconsin River. And you can go out there now and it's not perfect and it's relatively clean, especially compared to, back 30, 40, 50 years ago. But it's an interesting story in how a river has been able to recover, but you really don't want to dig up those sediments that are in the river. No dredging.
Yeah, that's a good example, I think, because I think we often, especially when we're talking about global issues, we tend to forget about what's right in our backyard. That's a good example. Thanks for bringing that up, Scott. So I have a quick question for you all as students. I'm wondering how many of you are savvy enough to have picked up on this and might be able to guess the answer. So Marie mentioned that when she was looking to determine how much mercury the birds were picking up in those particular regions, correct me if I misheard you, Marie, you said you were using the blood mercury and not the feather mercury, correct?
Yes.
So why would she be using blood mercury instead of feather mercury on a shorebird? Dan looked excited. Anybody other than Dan have an idea? Raise your hand if you know. See if I see anybody. Okay, so Katie, you don't count. Not a student. Okay, Dan, go ahead. Or you don't want to actually say it out loud. You just want to know privately.
I'll say it. I just wanted to give anybody else a chance. Looks like Charlotte might have.
Oh, did Charlotte? Did you raise your hand?
She's saying a thing.
She's saying.
Sorry,
a thing.
I typed.
Okay.
The only reason I'm aware of it is because I actually wrote one of my papers on runoff, and learned about it sort of, and just how a bunch of other stuff just kind of spreads through your blood, and then it will cause your organs to shut down. I know the liver is the big one. It attacks the liver first. The liver pretty much shuts down, and then you just die. So that was my only theory. Is like, well, because it absorbs through your intestinal wall quicker, I guess, and spreads. So.
Okay.
Is that
actually?
I have a question based on that answer. Is the liver the fattiest organ other than the brain? Because it's fat, right? It's lipids that it binds to, and it's like.
Not for mercury. So mercury, so that is true of a lot of contaminants and persistent organic pollutants like PCBs and many other contaminants. fat is where it gets stored, but actually that is not the case for mercury. It actually does not actually go in fat at all. Fat almost always has really low concentrations.
Weird.
Yep.
Yeah, so it's in what you eat, which is the muscle tissue. So that's why, I mean, that's in part why it's a serious concern, because I mean, when you eat meat, you're eating muscle. So you can't just cut the fat off and eat a lean piece of meat, or you can't cook the fat off because it's in the meat itself, in the muscle tissue. Yeah, it's a problem. It's a lot different than PCBs. So that's a good guess, I think, Charlotte. What were you going to say, Dan?
It was essentially that. I mean, I thought it was fat based, so I figured there are more lipids in blood than feathers. But it sounds like, is it protein? I don't know. I still don't know what it binds to. But if it's, I mean, there's more of that in blood too, right?
Mm-hmm.
Is it protein?
So that's a good question, right? So I may have mentioned that there's a lot of stuff we don't really know about mercury, and it's a really weird thing. So methylmercury actually mimics an amino acid. So it looks the same shape as one of the amino acids. And so that's why it can be transported really readily around your body, because your body just thinks it's an amino acid shape wise. It does bind, so it really likes sulfur groups, and so it binds to heme, it binds to hemoglobin in blood. But there's also like just some weird things that we don't really know what it's doing as it's kind of moving around. So there's a lot of so toxicokinetics, which is kind of looking at how mercury move or contaminants move in the body. That's one of the things I kind of focused on in my PhD, in birds particularly, but we still don't really understand how it moves around in humans either, which is something I kind of looked at in my postdoc and was like, oh my God, humans are the worst. I don't even want to know. And then went back to wildlife.
Humans are the worst. Dot, dot, dot. So
it's
there's
really complicated.
Yeah, and that's what I think is so, that's why I like talking to you about this, because it's so fascinating to me. There's some chat going on about feathers and how long birds hold on to their feathers. And so, Katie, what were you going to say?
Yeah, I was going to say kind of along those lines where feathers are going to be more of a historical record. Kind of like if you think of mammals, your hair can tell you a lot about the years of things that you've been carrying or been exposed to, versus blood is going to be an immediate picture. Feathers, if you're losing them every year and you're migrating like massive distances, you might not be exposed to it in your new environment, but it's an environment you're coming from.
What about that, Dr. Perkins?
That's exactly that. She's got it. And shorebirds are these long distance migrants, and they're also really weird molters. Like they just molt at really odd times, and people don't really, it's not well studied. So oftentimes we're not really sure where or when a feather was grown. But Katie had it that blood is a much more recent indicator of exposure, because it's going to be kind of circulating around your body more readily. Feathers are going to be kind of more of a snapshot of the time when that feather was grown.
Yeah, so I'm looking at Taylor here. You've got such long hair, Taylor. If you think about taking your hair, you can actually say, well, we know that human hair grows at such and such a rate. You can actually measure it out in inches and say, well, when this little bit of hair was grown, I was eating this much fish, and you can actually see the amount of mercury that you're eating based on your diet, based on what part of the hair you're looking at, which is totally creepy to me, you know?
Yep. But I actually had kind of meant to put in a slide on bioindicator tissues. So yeah, hair, and then one of the ones we were using more recently during my postdoc was fingernails and toenails.
Yeah.
Which are really gross.
Yeah.
We were looking at, this is like a random story, but we were looking at mercury in populations in Madagascar, and there's like a cultural thing with not giving your hair to people. And so they refuse to provide hair samples due to like their cultural beliefs. And so we were able to get fingernails from them, which was as disgusting as you might think, even though they're not really, because they're just on your hands, but yeah. Gross.
I was involved with a really cool study in Nebraska where we were having mountain lions showing up in eastern Nebraska that were continuing traveling eastward, but we were wondering where they're coming from. And so some of the mountain lions that were killed because they were in unsafe areas like downtown Omaha, we were able to take the fingernails, or claws in this case, section those claws and then look at the various sections and look at the isotopes of hydrogen, nitrogen, oxygen. And we could look at the background isotope levels, maps, and identify exactly where that mountain lion had come and gone. It was just the coolest thing.
Yeah.
So fingernails aren't that bad.
They might be bad. Yeah, so when I was talking about what mercury likes to bind to, it does like to bind to keratin. So it'll bind really, really quite tightly to both hair, fingernails, and feathers, which are all these keratin tissues. And in birds, molting is kind of this excretory method. Humans kind of excrete mercury pretty readily, but birds don't have a lot of excretory methods. I said they put it in eggs, females will, and then they'll excrete it during their molt.
So the feathers are basically like the dumping ground for toxins. That's
absolutely.
That's the mechanism.
Yep.
Never will look at feathers the same way again, I feel, right?
Nor fingernails.
Oh yeah, or fingernails, absolutely. Guys, it's 5 o'clock. I really appreciate you all being here for this. Are there any final questions for Marie? All right. I tell you what.
A lot of nice send-offs there from students.
Yeah, I really enjoyed it as well.